It's a pleasure to be here. I gave a talk on sudden of death. Who remembers when? At the national meeting in Berkeley, not the national, sorry, statewide meeting in Berkeley, maybe 9 years ago or 8 years ago. It was a long time ago. And so it's kind of interesting to be back talking to CNPS, even if at the chapters meeting. And the event that was not mentioned today is the upcoming Sawdust, so the citizen survey for this part of the world. And in fact, you're very lucky that here you actually have 3 different events, one in Atherton, you have one in Los Altos Hills, and then you also have one in Montalvo and one up in South Sky. Actually, you have 4. But I think that the chapter picked the one in Montalvo, and it's on the last Saturday of the month at 10 o'clock in Montalvo. And I hope that you're going to be able to join. And I will tell you a little bit of how important this citizen scientist effort is. But I will tell you more when you come to Montalvo. So we have been in the news again. We got good coverage by the Mercury News and Oakland Tribune about the effort. And so I'm very happy that there is some recognition that what the volunteers are doing is very, very important. And in fact, I will start with what I'm going to tell you at the end, that one of the interesting outcomes of the volunteer-based campaign, which is really extremely large. So last year we had about 600 people participating to the volunteer-based campaign, but what they produced was looked at by 1 million people. So 1 million people looked at the results of what the 600 people did. So it's really an effort that's having a significant impact on letting people know where the disease has arrived, and as you'll understand today at the end of the talk, knowing the fine scale distribution of sudden death is extremely important. It's probably more important than for any other forest disease that we know, with maybe some few exceptions. The first slide that I have here, it's a slide that I was attempting to show that the understanding of a disease like sudden death is very difficult and complex. There's a lot of elements. And what we did for the first few years for this disease, which is a completely new disease on the face of the earth, was to deepen our understanding of each one of these individual aspects one by one. And then about a couple of years ago, we started finally to be, we were able to start putting the pieces together a little bit. And so try to kind of provide a picture of what is going on, predict what will happen, and potentially figure out what is our best shot at controlling the disease. Let me see if I can figure this out. I think again, no. No, sorry. Okay. So just a few pictures to show you the different phases of sudden death. So and it's been very difficult for people that are not particularly tuned with the importance of individual tree species to understand the tragedy that really sudden death is. And what I would like really to start the main part of the talk with is kind of a statement that indicates that sudden death is not simply a huge ecological disaster, an environmental disaster, because of what it's doing to a coastal forest, but it's also really a political issue because of the way it came to be. It is a regulatory issue, and it is an issue that is international. In fact, it's a worldwide issue because of the origin of the organism, where the organism is found, and how it's moved around the world. So there's really a lot of aspects, and I will tell you some of my personal experiences. Because the entire forest doesn't die out, but only 2 or 3 species really die in very large numbers, the impact sometimes is not so visually dramatic as like the reporters would like it to be, for instance. They want to see the entire mountain brown. So a big fire, which sometimes maybe it's not as relevant as a disease like this one, gets a lot more attention, a lot more funding for research than something like this, because overall the forest will remain green. We have other tree species that will remain green, and overall we don't lose absolutely everything. However, the 2 species that are affected in very large numbers are 2 keystone species on the coast of California, Tanoaks and coastal live oaks. So Tanoaks really are the main, with Douglas fir, They're the main associates of redwoods. And the impact that the disease is having on Tanoaks right now probably is going to also affect redwoods in one way or another. In fact, some research already indicates that there are changes in the regeneration and in the growth rates and so forth. But it's going to take, decades before we can actually determine what the real impact on redwoods is going to be. Coastal live oak, of course, is the major tree in California for, the drought adaptation. So it's a tree that does very well on the coast where conditions are relatively messy. It does very well relatively inland when it's dry. It goes all the way from Baja to Ukiah, so it has a very, very large geographic range. And both species are the two main, especially Tanoaks are the main masting species. And so losing this source of acorns, the major source of acorns, especially in the case of Tanoak, has already had huge impacts on wildlife that uses the acorns as a down. Another picture here, this is Big Sur. So in this case, if you're lucky enough that you actually look at that mountainside at this particular moment, you realize how serious the disease is, right? So you can look at the number, you can estimate how many trees are being affected. And in fact, it's no surprise that in those areas, up to 100% of the Tanoaks, adult Tanoaks, have been killed by sudden death. So in many populations have already disappeared as adults. So they haven't disappeared as regeneration and shrubs because these plants are very prolific. They produce a lot of acorns, and it's going to take probably tens of years before those the small regeneration is also killed. But eventually, the prediction is that pretty much everything is going to go where the climate is very favorable or where the environment is very favorable for the disease. As the environment changes, and in particular as it gets a little bit drier, potentially Tanoaks can survive. The disease is not at its optimum, so we're going to have some fringe areas without the without mortality, total mortality, with some adults surviving. And that was Big Sur. This is Sonoma. So I wanted to let you know that pretty much wherever you go in California, you have pretty much the same type of impact, even if the impact in the redwood Tanoak forest and in the mixed evergreen forest, where coastal live oak or black oak are affected, is a little bit different in terms of magnitude. So the impact on Tanoak is much more significant, and the impact on oak is less significant, but that may be just an issue or a matter of time. So the epidemiology of the disease is very different on the two, and it takes more time for oaks to become infected. So potentially the two may actually almost equalize in the future. Not completely because of a few things that I will explain. From afar, the observation was of very large mortality, but if you walk into one of these forests, which I wanted to tell you it's very dangerous, by the way, you will see scenes like this, pretty much. A lot of trees dead standing, a lot of trees down. And for instance, in this part of Big Sur, I'm a mycologist, I study mushrooms. When I started working 10 years ago, the local people were just, Matteo, you gotta help us because there's no more mushrooms. And one of the other consequences of the loss of these trees is that Tanoaks are one of the largest ectomycorrhizal symbionts. And the only ectomycorrhizal symbionts, because redwoods, as you probably know, are not in symbiosis with ectomycorrhizae. So they don't support mushroom production. So we're losing that, and obviously this is also having an effect on nutrient cycling, on carbon cycling, and on fire behavior. And we'll talk about fire behavior at the end. So actually to look. The organism that causes sudden death is an organism that nobody had ever seen until we isolated it in California. But it belongs to a genus. The genus is called Phytophthora, and believe me, it's impossible to spell it correctly in the paper. Because the H's and the T's, I mean, it's impossible. And Phytophthora means plant destructor, okay? And it's a very appropriate name because there's about 90 species in the genus, and about 85 of these species are, or maybe 80 of these species are pretty good aggressive pathogens. And I put there some names, but it's, you don't really necessarily have to look at them. However, there's two that are particularly interesting at the very bottom. One is called Phytophthora cinnamomi, that is the equivalent of Phytophthora ramorum, except for it has an even broader host range, and it has caused huge devastations in many native forests, starting from central Mexico, where it affects white oaks, to the Jarrah forest in southwestern Australia, to the Mediterranean oak woodlands. And just recently, with no surprise, but until I arrived, nobody really had thought about this, because cinnamomum is completely widespread in agriculture. People have not really thought about it, but as we know, microbes don't know that the field ends here and then the forest starts there. And so actually now cinnamomum is a pretty big problem in California, and it's a pretty big problem in 3 ways. It causes 100% mortality of 2 species of manzanita, the Ione manzanita and the white manzanita in the Ione area, and that's an endangered species, so it's a relatively big deal. It's one of 3 or 4 species that grow on the serpentine soils of that area. So unfortunately, that's a problem. It's causing a problem in Southern California. You know when you hear that there's a lot of oak mortality in Southern California and there is a drought? Well, the main reason why there is the level of mortality is because the cinnamomum is pruning the root systems. And so every time water supplies are lower because there's a drought or because there's the nth golf course that's being developed in Southern California, then the result is that as the water table goes down, because of the lack of fine roots, these oaks die out. Okay, so that's kind of one of the major reasons. And then, if you have looked, sometimes you see madrones that look really crappy here in the Bay Area, or manzanitas, or even California bay laurels. That's also caused by cinnamomum. But the big difference, lateralis, I wanted to mention the lateralis is, both cinnamomum and lateralis are exotic to California, very present in California. Lateralis probably comes from Taiwan, and it's the close relative to the sudden death pathogen. So that's interesting because it gives us some indication of where the sudden death pathogen comes from. Cinnamomum comes from Papua New Guinea. Okay, so they're both exotic. The big surprise when the sudden death pathogen was discovered was that it was the first Phytophthora species ever to be discovered in the forest system that was not water-based or soil-borne. Okay, so this particular organism, and the 2 of us that were working on it at the very beginning, we started saying that, wait a minute, this is not in the roots, it's not in the soil as it's supposed to be, we find it in the leaves, we find it on the trunk. And honestly, nobody believed us, because we didn't have a lot of experience working with these organisms. These organisms are not fungi, they belong to the same kingdom as kelp. Okay, so they are in the straminopiles, or Straminophila, and the brown algae, the kingdom of the brown algae and diatoms and kelp are included. So indeed, they are water-based organisms. And until Phytophthora ramorum was discovered, the general epidemiology for instance, cinnamomum and lateralis that I just mentioned, was mostly of an organism that infests the soil, moves through movement of the soil, moves through current in streams and waterways, and so it mostly infects the lower parts of the tree, sometimes for both of those species with catastrophic consequences. But this is not the way ramorum is. So ramorum is actually airborne. So it has evolved to be able to do very well on the aerial part of the plants. And this explains a little bit why the explosion of the disease, so the epidemic outbreaks, has occurred. Because once you're aerial, you have a lot more potential to just grow, you have more potential to spread, because all of a sudden you don't have the barriers that are put forth by the structure of the soil and the limitation of soil movement and water movement. You're basically free to do whatever you want. It is. I would like to say that it's still a water-based organism, and what I put right there is kind of the key to understand the biology and the epidemiology of sudden death. So there is a phase in the disease cycle, and I'll show you the details, where swimming spores are necessary for the infection process to be effective. So that phase is absolutely still linked to water. Okay, now because it's not in the soil, so it can be water in the soil, it cannot be water in a river or in a lake or the water table. The water that the organism uses at this point is rainwater. And we were a little bit surprised because we expected that California would have been fog as well, right? Especially because the organism is very, very widespread in the fog belt, or whatever we want to call it, in the coastal area. That's not the case. For some reason, it really requires rainwater. So the infection cycle, the levels of infection, and the geographic spread of the organism are absolutely correlated with the amount of rainfall that we have in California. Okay, so you understand immediately that it's not always on the go. Sometimes it stops because we have plenty of droughts and plenty of dry years. Fortunately for the organism has been incredibly good at evolving, thinking about everything. And what he thought was that, wait a minute, if I don't have water, what am I going to do? And through evolution, so it happened that it produces a huge amount. In fact, ramorum probably among these 80 species is the species that produces the largest amount of very thick-walled spores, and these thick-walled spores allow the organism to survive at sub-zero temperatures. We're talking minus 50, minus 60, and it allows it to survive in desert temperatures, so very, very hot temperatures. However, once it goes into survival mode in the structure that are called chlamydospores, all of a sudden, it starts being active, it stops being active. Those chlamydospores basically only are important for the survival, but we think that they don't really play a role in the epidemiology. Okay, so the epidemiology, which means actually the infection rates and so forth, it's a different story. Chlamydospores are slow in waking up, and so they're not really participating in the yearly infection process. However, they allow the organism to survive. So, because of this, we're basically never going to get rid of, we're never going to get rid of Phytophthora. I also wanted to let you know that it was interesting because when we said, I was the first person who said it's aerial, and people told me, Matteo, you always talk, you don't think. And after that, we discovered six other species of Phytophthora that are also aerial. So we opened a huge can of worms, so now all of a sudden we know that it wasn't true that there were no aerial Phytophthora. And this is kind of interesting to us because in the Bay Area, actually throughout the range where ramorum is, there are at least 2 other species that are relatively abundant and that are potentially competitors for ramorum. So we have noticed that especially if the conditions are relatively dry, so if it's not a very wet year, in some locations, in San Mateo County for instance, these other aerial species can pretty much match the same infection rate as ramorum. Okay? But unfortunately what happens is that start the rain, ramorum goes up exponential and the other ones just go up a little bit. So we know now that really the ability of ramorum to explode in terms of population size within 48 hours from the first rain event is what makes the big difference. The other species just simply cannot keep up with it. Okay, so the first, for those of you that don't know or may not remember, the first reports of sudden death probably come from the 1994, 1995. So we have some written reports that Tanoaks were dying. And that was unfortunate that it was Tanoaks dying because it slowed down the entire process because at that time, Tanoaks were thought of to have no importance. They were thought as invasive shrubs. They were competing with the timber species. That was the mentality, okay? And I must say, very wrong mentality because of the ecological role that Tanoaks play. And all we had to do is just simply look at what the native people of California thought of their trees. And on the coast of California, the sacred tree is not a redwood. The main sacred tree for the native people is Tanoak. And that's because it provides the acorns. And the second most sacred, almost every tree is sacred, but if you prioritize to the native people, the second tree apparently is bay laurel, which is interesting. The species was identified as a new species in 2001, and so everything that we have learned about it, we've learned in about 10, 12 years. And the number of people that went from 2 of us, we at Berkeley and Davis studying it too. I think now we have like 600 people that are working almost full-time in the world on this organism. It is considered the most dangerous pathogen in the world for trees. Okay, so it is also the most highly regulated plant pathogen in the, for all plant pathogens. It is not native to California, and this is the main problem. We don't really know where it comes from, although I told you that its close relative comes from Taiwan, so we have a strong suspicion that it may come from East Asia or maybe from the Himalayan range, and I will tell you more about that. But the most important thing to realize is that not only once it is spread in California successfully, it finds the native flora without any ability to resist, which is, we all know that, okay? So it's, it's the usual story of an exotic organism to which the native flora has developed no resistance because it's never been exposed to it. But there is other parts of the story or other pieces of the puzzle that are equally important. The first one is that because it has arrived recently, in fact, we think, if I had to put a date, I would put 1987. That's when I think it escaped into the wild. And that's because it was an El Nino year. And 87 gives enough time for the 1994 report to actually have occurred, okay? So, take my word for it. It's 87. It doesn't matter. Anyway, it's a pathogen that's very aggressive, so it has arrived relatively recently. So what is it doing now? It's still expanding. It hasn't occupied at all of the areas that it can occupy. And in fact, I left this at the end, but I'll tell you now, as of last year, it has only occupied 15% of the range that it can occupy. So this should be a kind of warning that, even if we're becoming a little bit complacent and we're fatigued by sudden death is, just at the beginning of its life in California. It still has 85% of favorable habitat to conquer. And the other, and even more disturbing aspect is that it constantly expands its host range. Why? Because as it moves in different areas, it encounters new plant species, and with these new plant species, it starts new relationships, normally pathogenic, different levels of pathogenesis. However, this is very disturbing because we don't know what the potential is on all the plant species that it may encounter. So one of the things that we really are trying to avoid is avoid excessive encounters with, let's not take for granted that it's already here and we know what it does. We don't know what it can do. We don't know. So the least it encounters, the better it is, okay? So if we keep it at just to do what it's doing right now, it would be a very good thing. So the reason why we're concerned about sudden death is, of course, because of the mortality of a few tree species. But you should know that as of today, the sudden death pathogen can infect every single plant species that grows on the coast of California. From herbs to monocots, grasses, pretty much everything can be infected if it is in the right habitat. Okay, so it has a really broad host range. However, I don't want to paint this doom and gloom. It's the effect on most of these plant species is negligible. It doesn't really do very much. Okay, so we don't really have to worry. Well, at least at this point, it seems to be negligible. The big victims are oaks and Tanoaks. The most important host for the disease is California bay laurel. Okay, so actually the organism was brought into California, and I'll say how now, and when it escaped in the forest, it escaped to California bay laurel. And it currently is using California bay laurel as the main vector that spreads the disease around, around the coastal forests. Unfortunately, California bay laurel is not really affected significantly. It's the most infectious host, but it doesn't even know it's infected. And so there is no feedback mechanism. And in fact, if you read some old literature, the old literature said that once the oaks die, there's a transition to the Douglas fir. You probably know this, right? In theory. Well, now with sudden death, we have to change that because what happens is actually the really large number of trees dying don't really allow the Douglas fir to come back very easily. It's bay laurel that comes in. So we actually are increasing significantly the percentage of bay laurel, so things are going to get worse. Of course, bay laurel, 100 years ago, there was probably at least 40% less bay laurel than there is now because of fire exclusion and different practices that we're currently employing. We favored bay laurel tremendously Of course, without knowing that, 50 years later, a little microbe called Phytophthora ramorum would have arrived, and that this microbe would have used California bay laurel as its main vector. So it's sad, and how sometimes we should really try to avoid these human-generated changes as much as possible because we don't know what the implications of them is. If we had less bay laurel, we would be in better shape because it would be like having less mosquitoes spreading malaria. The organism arrived in California through the ornamental plant industry. It affects, I think, 75 or 80 ornamental plant species. However, the three most important ones are the rhododendron, camellias, and viburnum. And in fact, we think rhododendron is a very likely candidate for the species that brought it over for a variety of reasons. So originally, the disease was in nursery plants exclusively, in a few species because there was cross-infection within the nursery. It was not in the wild. And it became very pervasive in the 80s throughout the ornamental plant industry, both in North America and in Europe. Okay? Unfortunately, this escape, occurred mostly in California because of the nature of the residential developments in California are very different than in Europe, for instance, where there is more separation between the residential areas and the wild areas. On the coast of California, it's very common to have the residential neighborhoods right in the forest. Maybe the forest is cleared out, but then it's planted with, basically, the people live kind of in the forest, and this made things really easy for the escape of the organism. There were two types of escapes, okay? So one escape was directly from nurseries into the wild. And that was fairly important for you guys here because your infestation here comes directly from Santa Cruz, probably a single nursery in Santa Cruz County that started infestation before anybody else without knowing. The second method, the second known pathway of introduction of the disease was through ornamental plants that were sold, right? So individual people were buying ornamental plants and they were planting them, and if everything went well, the organism was able to escape and to move in the forest. So because humans move a little bit wherever they want, as far as they want, the initial introductions were a little bit everywhere, and I'll show you a map a little bit later. From this original introduction, the organism moves by itself, and it moves through when we have rainfall and using the wind. Okay, so it's really a wind. It uses the wind to move. It does need the rainfall, and the infection structure is so gigantic that the average movement of the organism doesn't go over 10 yards. So it is extremely limited in its ability to move. However, there is a trick. The organism produces so many gazillion spores that even a very small percentage of these makes it for a longer distance, then you have transmission to, for instance, a different patch of the forest. So that happens, but that doesn't happen very frequently. So mostly what happens is that the disease spreads locally through continuous or very nearby patches of forest. So that's what happens normally, especially if the conditions of rainfall are slightly below the average, like for instance in this year. Okay, so that's the common pattern, and then occasionally you have the longer pattern. So this is established. Plants introduce the organism, then the organism moves by itself using the wind. However, through laboratory experiments, we can prove that two things are infectious. Well, water actually is infectious. So in the areas that are infested, the water is infectious, the soil is infectious as well, and of course, the green waste is infectious. So if you cut down, bay laurel leaves or madrone leaves and so forth, those are infectious as well. We don't have any evidence that these, the soil, water, and green waste has moved the organism. We have maybe one or two instances where we suspect. But the other two really explain 99% of what we see. However, from a regulatory perspective, we need to be cautious no matter what because we don't want to enhance the spread of the disease. Nonetheless, I also have to tell you what the truth is about about sudden death. So, this is what the organism looks like in a petri dish. It's kind of cute, actually. You know, it's funny because, of course, I was the first lab to have it, and I wrote a letter to, I sent a, you know, a copy of this petri dish and a letter to Washington telling them we isolated the organism that's responsible for sudden death, you know, given that we haven't done all the experiments. And of course, the FBI comes in the lab 2 days later saying that I'm in violation of federal laws because I have a very highly regulated organism in my lab. And I'm like thinking, wait a minute, I discovered it, so then I give it to you, and then you want to put me in jail because I give it to you? I mean, so this is the interesting, you know, sometimes our government doesn't really think. So because the organism is new to science, the situation of sudden death is different from other exotic diseases. So for instance, chestnut blight. Okay, chestnut blight was a huge scourge, you know, for chestnuts, but the organism was already known. And it was known where it already existed. So it's still an exotic organism, but there is some knowledge that you can actually rely on. In the case of sudden death, we didn't know anything because we didn't know anything about the biology, the origin. We didn't know anything about the symptoms. And one of the problems was that it became, you know, level 3 quarantine organism, and it's also considered a biological weapon because of what happened September 11. So that makes research particularly difficult because of the limitation imposed by the federal government on the organism. So one of the things, this is what, you know, these were the first pictures that we took of the organism. So do you remember those spores that allow it to survive when it's particularly hot or cold? These are them. And in fact, this is the very thick wall. You can see it as a light layer on the outer part of the sphere. The most important structures are the ones on the left. Okay, those are the sporangia. And those are the structures that are gigantic. So they're 50 to 70 microns in size. So they're really, really big for the microbial world. And they have several features that make them very effective. They detach themselves from the stalks, they carry them, and then they become airborne and relatively pervasive because of the fact that they can detach themselves. You should think of them as pods, spaceships. So they are like spaceships, and they go from, you know, plant A to plant B, which for the microbial world is from planet A to planet B, right? In relative terms. Once the spaceship arrives on the other plant, if it lands on water, what will happen is that a hatch will open, literally, and the hatch is on the tip, and 20 to 30, but mostly 30 spores will swim out, and they will swim, all of them will swim towards the host, and they will infect the host without the need for any wounds. So these are so tiny, they're propelled by tails, they absolutely require water, and they can go through the stomata or through the lenticels. So that's kind of the reason why infection is so effective. But it is under this constraint of the presence of a film of water, and I'll give you a little few more details. So that organism that I show you right now, right, on rhododendron plants, and the problem on the rhododendron plants, and the symptoms that it causes on rhododendron plants, is that the symptoms are totally generic. I mean, who hasn't seen blotches on rhododendrons and spots and wilting tips? I mean, it's the most common symptom you can have. There was no way that people could have even figured that these ornamental plants were carrying this really dangerous microbe. On top of that, rhododendrons have a unique feature that no other plant that we know of has yet. The organism can stay in, it becomes systemic in the plant, but without showing any symptoms. So for several months, the plants look perfectly healthy, which is perfect for the ornamental trade because it allows the plants to be moved without any worries. So that's why we really think the rhododendrons are probably the most likely candidates for the arrival of the organism. And this is the first philosophical question. Why do we need to take cuttings of flowering plants from all over the world? And why do we need them to bring them to different parts of the world, spreading the microbes that they carry? I mean, this is really not necessary at all. I mean, we don't need these plants to survive. Well, I started voicing this, idea, and the dean of the president of UC Berkeley calls me in Oakland, and he shows me a big book, The Economy of California. And the second line of The Economy of California for the agricultural section of the book is the ornamental plant industry. So it's the second most powerful industry in the state and in the country. And it's not mystery that they hate me because I was the one that proved that they really brought this organism. Nobody necessarily blamed them, although we do blame the practices. So both in Europe and in North America, and I'm sure you're familiar with this, the large companies, let's say Morovia and so forth, they figured out that it's much cheaper to transport plants than to have different growing beds in different states with different environmental conditions. So all of the plants that are sold, in these large facilities, are all grown in California and Florida, all of them, for all of the states of the United States and for Canada. So what happened is that the organism got into some of these networks, and once it gets into the network, it's going to be spread everywhere. In fact, sudden death, when we were studying it already, and that will be the second philosophical point that I made, was transported to 22 states because of these practices and because of the way the country, and actually every country in the world, was regulating plant material. So their approach was faulty. And we've proven them wrong immediately, and as a result of that, the sale of, I don't know, millions of infected plants, we now have the pathogen in streams in about 6-7 different states in the East. In the streams, it hasn't come out of the streams. There was a huge recall, but so it's out there, and sooner or later we'll see it. We'll see it out in the forest. So in 2001, we started trying to figure out where the disease was in California, because we had a few reports, one in Marin and one in Santa Cruz County. That's it. And so we said, wait a minute, it's probably more than just in Marin and Santa Cruz County. And to our dismay, we figured out that it was already where you see all those red triangles. So in 2001, it was already there. So we had kind of missed the train, missed the boat, or whatever you want to say. We arrived a little bit too late. The organism had already spread to, pretty much almost kind of the entire range that it occupies today. In fact, if you look at the distribution a few years later, 2007, it is not all that different. Okay? The only big difference is Humboldt County. Okay? So Humboldt County came 2 years later, but everything else was pretty much already there. What has changed every year? It's the coverage in the local areas. So it has increased exponentially. Okay? So that's what it has changed. So it's not that it's not moving, but remember I was telling you it doesn't move all that fast. And in fact, this is what, in a paper that we published a few years ago, and one that's coming out, a mega paper that's coming out now, we were able to reconstruct, and this was not easy, we were able to reconstruct, based on our sampling, which of course is limited because you can't sample everything, all the locations where we could prove, and I won't be tedious and tell you how, we could prove that the organism had been introduced by humans, okay? And those pink dots are placed exactly in those locations. So you can see that those pink dots pretty much explain most of the range. So again, the organism was being introduced simultaneously in many different locations. People didn't know, of course. And then it's been spreading, we think, mostly naturally. Or at least we can't discern whether it was natural because the range of spread is too similar, and we can't really prove or disprove that humans were moving it because it's still within the potential movement, natural movement through the air. This was a cornerstone, a turning point in our understanding of how the disease moves, because until we could prove with confidence that that's exactly what had happened, we had this sneaky suspicion that the organism really moved hundreds of miles in a few years, not necessarily naturally, because we can see that it didn't move very far, but potentially maybe people were moving all the time. Okay, so we had a sneaky suspicion, and so we were in between thinking of very complex ways of addressing the issue. And when we found out that this was the most likely interpretation, it's a little bit better. It's a little bit better because it means that it doesn't have this huge potential, whether naturally or through human development. And fast forward 10 years later, and this is a little bit, just a little bit of tidbits of scientific information just to keep you on the edge of research. So what we found out was also very interesting. We found out that actually the two things, and this is when the nursery industry basically tried to put a bullet in my chest, because we published a paper where we showed without any doubt that because their statement was, oh no, we got it from the forest. You cannot prove which way it went, right? We got it from you guys. It's your fault, right? So they were using the opposite argument, and we were able to show that it was basically impossible, that the strains that were in the nursery, for two reasons. And one is very simple and intuitive, and it's incredibly important for California. The nurseries actually have four, not these four, there are four completely different types of Phytophthora ramorum, okay? Completely different. The forest only has one. So, evolution tells me that it is impossible to go that way, it has to go the other way. So, from 4 you can go to 1, in 10 years, but you cannot go the other way around, it's basically impossible. So, we're talking about groups of strains that took hundreds of thousands of years to develop. Okay, so the nursery has this in its circulation. California only has one of those four. And on top of that, the strains that were found in nurseries were always the great-grandfathers of all the other strains in California. So no matter what type of genetic calculations, genetic reconstructions you do, they always come as ancestral. So we know that the ancestral strains to the entire epidemic in California come from the nurseries. I wanted to point out that those are what I call the one, because this is something interesting we don't understand fully. You would expect that the group number 1 would be the most prevalent in California because it was the one that was first released. It was the one that got the jump start. But it's not so. It's actually the least prevalent. So we don't, I don't want to tell you stuff that's incorrect, but one of the hypotheses is that potentially, these organisms can evolve very rapidly, that potentially there was a change that makes them better in the forest with respect to the strains that are in the nursery. And now we have 4 groups of strains, and it's interesting that they are geographically separated. So the Northern California has one, Big Sur has another one, the Bay Area is one, and then Santa Cruz County and a few areas where nurseries are, I have the original one. So it's kind of interesting that this is going on. By looking also at the genetic structure, not just of the 4 groups, but within the groups, we were able to prove the concept that the organism doesn't move very far. So we were able to calculate over and over again the number of migrants. So the number of individuals that go from one forest to the next. And we were able to show that it's very, very small. Okay, so that there's very little migration, it's very infrequent, and that supports the idea that the organism doesn't move very much. Okay, so I already explained the major concept. The second one is important here in the second bullet point because of the patterns and the mechanisms that I explained. You have to think of Phytophthora ramorum not as a blanket throughout the state, but it's extremely patchy depending on where it was introduced, depending on where the habitats are, and depending on where it was able to reach. So it's incredibly patchy. There's many, many pockets that don't have the disease, okay? And so this is very important because in our calculations of the overall infestation rate or infection rate, patchiness makes a huge difference in terms of the end result. And it also makes a huge difference if you are in a pocket that doesn't have the disease versus a pocket that has the disease. And that could be just in the scale of 1 kilometer, okay? So less than a mile. So again, not everybody that lives even in a neighborhood, well, a neighborhood maybe yes, but you can be in a town and you can be on the same side of town and have 2 completely different situations because of the history combined with the habitat. Okay, so frankly, where did they arrive first? That area may have it, the other area may not have it, and then it's going to be compounded potentially by the lack of habitat or the distance between two favorable areas. And of course, there's some counties in California on the coast that don't really have a lot of the disease, and it's not surprising. San Francisco, I don't know why, Solano and Lake counties. These are counties that are coastal. So every coastal county from Humboldt to Monterey has it, including the bay, all the counties around the bay. But those three are either, urban or at the edges. And that's where the habitat transitions into the interior habitat. And the interior habitat, it's not as favorable to the disease. The organism can still survive, but once it moves there, we're not going to see sudden death the way we know it, because the epidemiology is going to be much slower, it's going to be much different. So the symptoms, bay laurel is the major host of the disease. Those lesions that you see there are there because water will accumulate there. And remember that you need water for the infection process, and you don't need water just for 10 minutes. You actually need water for quite a long time in order for the infection process to be completed. It is scary to think that within 48 hours, the organism goes from infection to megasporulation. So it can produce hundreds of thousands of those pods that I showed you earlier on that are called sporangia within 48 hours. So it's incredibly, incredibly prolific and incredibly fast. And this is the key to its success. That's why it's such an aggressive organism. In schematic terms, this is how the epidemiology of the disease is. Okay, so on a yearly basis, as long as we have a little bit of raid, we have infection that compounds itself on bay laurels, both within the same bay laurel and maybe on adjacent bay laurels. Okay, so this is what happens on a regular year where we have, if it's a very completely dry year, we don't even have that. Okay, but normally we have a little bit of rain. And this is what I was telling you about the hatch opening. You see the hatch opens, and you see these dots are actually the spores that are squeezing themselves out, and then they start swimming. Okay, so that's the phase that requires water. This goes on and on and on. When we have a wave here, so when we have above average rainfall in the spring, because the organism likes warm temperatures, that's when the pathogen will move from bay laurels to oaks. In a dry year, it will never infect oaks, okay? And the reason for that is that, believe it or not, oaks are really hard to infect. Tanoaks are a different story. Okay? So Tanoaks are very easy to infect. So Tanoaks behave a little bit like bay laurels in terms of infection. The difference is that, unfortunately, while in bay laurels there is no infection of the wood, of the cambium, nothing, just the leaves, in Tanoak you also have these secondary infections on the trunk, and those are the ones that end up killing the tree. Tanoak leaves are also infectious. So the leaves of Tanoaks and the leaves of bay are the major vectors. And of course, the leaves of Tanoaks being close to a Tanoak are very easy or very convenient source of inoculum that will infect the Tanoak and kill it. So Tanoaks, unfortunately, every year the mortality rate increases. But oaks, no, because of the large number of spores that need to arrive on an oak for the infection to happen. Very large, I mean, we're talking 10 to the 5th, okay? We need 10 to the 5th spores in order to actually be able to infect an oak. How does oak infection proceed? Well, it goes under the bark, it gets to the cambium, and then it very rapidly eats the cambium of the plant. The plant is not able at all, almost no plant is able to replace the cambium. The moment that happens, the plant is basically dead. It can happen in as little as 1 month, okay? Well, my first research project was on chestnut blight, and we thought it was fast. This is much, much faster. However, you don't see the effect of the girdling for many, many years. So the plant can be dead after, 1 month is the fastest possible. It could take up to 2 years for the girdling to occur, okay? So, just to be clear. However, the plant doesn't die right away. It stays green because these are drought-adapted plants. They have tons of resources, and they use all their resources, and you know what they do? They put out flowers, they bud, they produce acorns, and people are happy under their oak because they think it's alive. And all of a sudden, the resources end up, and all of a sudden, the tree becomes brown, and that's why it's called Tanoak death. But that tree was dead for many years, because that tree was in fact dead when it was girdled. And, but we as humans are stupid, and we don't understand that the green tree that was girdled was dead. We think it's still alive because it looks green.
What eats the cambium?
Huh?
What eats the cambium?
Phytophthora, the organism itself grows and eats everything that's sugar, starches.
Vegetative organism?
The microbe. The microbe itself, it's what the microbe uses as food. Okay, so, and in the process of eating, it destroys the cells. So this, oaks are not infectious at all. Okay, so they don't spread the disease. So it's a first plant pathogen where all the models, including I do a lot of genetics, besides population genetics, the only models that work are the models of zoonotic diseases like malaria, West Nile virus, where you have a vector like mosquito and then you have dead end hosts. So these are the models that work for Tanoak death. It really behaves the same way. So and this has lots of implications, and maybe a more advanced talk will talk about that. The good news in this sea of tragedy is that not every oak species is infected. So really this is a problem for the red oaks. Okay, so California coastline oak, black oak, and Shreve's oak. Okay, and it's interesting because these three oak species, even if black oak is deciduous, are they're the three closest relatives. Okay, in the clade of red oaks, so they're very, very close. So the disease has nothing to do with a plant to be deciduous or not. Unfortunately, canyon live oaks also seems to be susceptible, but to be honest, I'm not sure that we fully understand what goes on. And here is a cross-section that shows you what happens. So the black line on the outside is the dead cambium, but as you can see, it also goes inside the wood a little bit. And so actually, the mortality is due to two things. One, the girdling, so the killing of the cambium, and the second aspect or reason for mortality is the blocking of the vessels. So both things are going on, and this accelerates the mortality. Tanoak, completely different story. As I said, in Tanoak, look at the size of that canker. We inoculated that Tanoak at the very beginning of our studies, and that was the size of the lesion like 40 days later. So Tanooks are incredibly susceptible. Tanooks get hundreds of lesions everywhere because any spore that ends on the trunk can infect it. Remember, in the case of oaks, that doesn't have it because you need a lot of them. And so basically, they're under attack. Plus, they spread the disease. They're completely under attack, and so that kind of explains the higher mortality rate. This is what a leaf that's infectious in Tanoak looks like. Completely different pattern from bay. Remember, in bay was where the water accumulates. Here, it's a little bit here it really likes the midvein and the petiole and the twigs. And so the organism will actually preferentially colonize those areas. It seems that it doesn't colonize the mesophyll directly, so what happens to the mesophyll is actually the result of drying because the midvein and the petiole are occluded. It seems that it doesn't like to colonize the mesophyll because of endophytic fungi that are there. So the fungi that are there are kind of saying, we're not giving it to you, we're holding on to it. And so the organism really is limited to those areas, but unfortunately, that's enough. So the tree is girdled, the tree is dead. We don't perceive it as dead. We're the only ones that don't perceive it as dead. Every other organism in the forest knows that the tree is dead. And these organisms, the smartest one, of course, being a mycologist, are the fungi. So the fungi, the wood decay fungi, most of them are endophytic in any tree that's already woody, So they arrive during the growth of the tree, when the tree becomes, woody from herbaceous, and they infect the tree, but they don't move. They're just there, They stay still for hundreds of years. When the tree starts declining, not just for Tanoak death, for any reason, what happens is very simple. With the decline, there's loss of function. Loss of function means that there's no circulation, and what gets in? Oxygen. So trees are very anoxic inside, so there's no oxygen inside. The level of oxygen in trees that are healthy is minimal. Once a tree is not healthy, oxygen starts getting in, and that's what the decay organisms smell. They breathe, they start growing, and as the result of the growth is that you have decayed wood and then you have fruit bodies on the outside. The problem is that these green trees can fall like brown trees, right? So because the decay processes start when they're green, the tree is green, but it's completely decayed and it can fall. And this is the first time that we have observed this. I mean, it's a phenomenon that probably has happened before, but because of the numbers now we have seen it, and so that creates a problem because obviously there's been several lawsuits of people being killed because trees are falling down green, and so it's a complication. The other group of organisms that is smarter than us are the bark beetles and the ambrosia beetles. These are tiny beetles that can only colonize for reproductive reasons trees that are very severely compromised. Okay, so despite what the entomologists want you to believe, these beetles have really no significant impact on the mortality of the tree because they can only go on trees that are almost dead already. What they may do, they may slightly accelerate the process, no doubt. But there is not a single healthy tree ever in the history of oaks being reported attacked by these beetles. So the reason why I have that picture is frass, because the beetles, they have relatively tiny entry holes that you can see, but it requires a lot of attention. However, as they're digging their tunnels, they produce a very large amount of sawdust, frass, that's kicked out. And so you will see the sawdust on the outside of the tree, and I'm sure that you're familiar with that. The sawdust is a good thing because it's a very good warning sign, hey, the tree is a hazard because the tree is very severely compromised, even if it is green. The same thing for the decay fungi. So those are things that we should treasure because they actually help us to identify trees that may be hazards. So, in summary, without spending too much time, we obviously have two situations in terms of mortality. We have Tanoaks that are affected in very large numbers, complete demise of some populations, all size classes can be infected. However, the game of numbers is such that the regeneration is so gigantic that in terms of percentage, the small sizes are not that severely impacted because there's just so many of them, okay? In the case of oaks, as you can see, the mortality levels are different, although that's wrong now. Actually, Tanoak is 100%, sorry, this is an old slide. It's 100% for Tanoak, it's 60% for oaks. So this was done previously to the last years. And interestingly, the small trees are not affected. So trees that are under 10 centimeters in DBH are never infected. And the reason for that, we think, is because water doesn't accumulate on the thin bark. It just kind of dries out. And as a result of that, the swimming phase can't really be completed. So as your CNPS, I want you to see the confirmed host list. And we could spend a long time looking at this. And these are all the native plants in California in that host list. But this is, again, this is from probably, I picked it 2 years ago, so there's probably 10 more species in this list. So the symptoms on each species are different, okay? So when we were discovering at the beginning, then we got really bored and we let other people do this, but when we were discovering the symptoms, it was just crazy because, like for instance, in California Buckeye, there are shot holes, look at there on the left, just somebody shot the plant with a little gun, and then the shot holes coalesce into a big lesion. Something completely different from what I showed you earlier on bay laurels. In maples, it's a scorch. Always a scorch, so again, completely different. In redwood beetles, it's a little lesion of the needles, okay? And I wish I put the Douglas fir one. Douglas fir is a wilting of the tip. So the symptoms are very, very different on each plant species. And this was the first problem for the regulatory world. Well, how do you train your people that are looking for diseases that are being imported or moved around to know what the symptoms look like if the symptoms look different on every plant species? So it's kind of a little bit of a problem. But there was a much bigger problem, and unfortunately the format of this is a little bit not how I wanted it. What I wanted to show you was the differences in results between the way all of the world was doing things before we arrived and the way they're doing it now. So before we arrived, all diagnosis of all plant pathogens was done through culturing. So legally, you didn't have the power to stop a shipment unless you had a culture of the organism growing on a petri dish. And this was all obviously a very pro-grower policy because we know very well that not always organisms kind of grow. But with ramorum, because it was so important, when we started seeing these differences, so the light green is actually what we could get by using DNA assays. And the blue, the dark blue, is what we were getting using the official government-based assay, not just for the United States, for the entire world. So I started a crusade to get things changed. And it was a long and painful process. But in 2004, in a meeting in Washington of the Secretary of Agriculture, a very heated meeting in the House, closed doors, but thank God I had access through people that I knew. And we were able to actually convince the government they needed to switch from culturing to DNA-based assays, like it's done for the veterinary sciences, for the human sciences, because the consequences are way too big. And that decision basically changed policy for the United States. 2 weeks later, Canada. 2 months later, the European Union, China, Australia, New Zealand. So now the entire South Africa, the entire world now has changed their approach. And so we, as Californians, have changed the way policy is made in the world. And that's not so bad. So that's one of the good things. Unfortunately, we had to be the ones that... Okay, so I told you this already. Sometimes I have the summary slides about the process of death of the trees. This is what I wanted to show you. This is very interesting because it explains to you why a certain amount of rain is important and why spring rains are important. So the first graph there without the axis because of the formatting shows you the size of the infection by the hours, okay? And the first one is actually, the first dot is 6 hours, the second is 12, 18, 24, 36, something like that. And as you can see, at 6 hours, so 6 hours, I mean 6 hours of a constant film of water on the plant surface. Look how small the infections are, okay? But look at what happens at 12 hours. So really, in order for infection to be very effective, it needs, and remember that with these infectious diseases, it's almost never plus or minus, it's probability. What is the probability, what is the likelihood? So when we get to about 10 hours, that's when we see a significant amount of infection. So that's what's needed, okay? So we need to have 10 hours of moisture for infection to occur. And this is bay laurel, so I don't know for oaks, but this is for bay laurel leaves. And the temperature needs to be around 69 Fahrenheit. And on the coast, 69 Fahrenheit usually is not reached for a few hours until April. So that's why normally you need to have rainfall in April, May, and June when we have it for new infection to be significant. Those years, if you have rainfall in those months, significant rainfall are the wave years. So 60% of oak mortality has been reached in California only through 3 waves. 2000, 2001, 2005, 2006, 2010, 2011. So now this puts things in a different perspective because Tanoaks have been dying every year, they go up to 100%. But oaks have only died in these 3 instances, have been affected in these 3 instances, and still we are at 60% mortality. However, remember that the number of sporangia is essential, so as oaks are more distant from bay laurels, and as the weather changes, the conditions will be better for oaks, so there's still lots of caveats. This was an experiment that we did in Santa Cruz County to actually nail down when these infectious spores, nobody wants them, they're dangerous, they spread the disease, and until we started the study, the only way we could measure that was to collect rainwater. We collect rainwater and then we would measure how many of these were in the rainwater. And there was a little bit of a problem with that because if we're measuring spores based on rainwater, then we're assuming there's none when there is no rain, but that assumption is not based on data, it's just based on the fact that we're not able to collect them. So what I did, I put 125 traps in Santa Cruz County, and for all those years, actually plus 11, and I think we did 12 too, I had every 2 weeks my students bring the rainwater to each site and bring the water and put it on the site for, And after this experiment, I lost all of my students because they had to hike miles and miles with gallons and gallons of water. And we were able to show that indeed, we only saw spores. So those curves that you see there, it's when we saw spores in these forests. Okay, that's the average of all the sites per year. So you see two things. They're all between March and June, and these are the infectious ones. So the spores that actually are produced in January and February are not infectious, okay? They don't really participate in the epidemiology of the disease as much. It's never a complete negative. And the other thing that you notice here is that the height of the curves is different. And guess why? Because of the amount of rainfall. Right? So years have different amount of rainfall. So we were able to, this is a nice close-up of the sporangium. You see with the spores coming out and swimming. So one of the things that surprised us a little bit, and we're going now on the plant side of things, which I think you will appreciate, was, okay, so if oaks are so difficult to infect, and in fact experimentally we just cannot infect oaks, even putting, you know, a lot of spores on the oak, and you just don't want to be infected. So why is it that there is this huge epidemic on oaks if oaks are so difficult to infect? And we came up with this theory, and we were able to publish it in Phytologist. It's kind of convincing. It's not foolproof, but it's convincing. And basically what has happened in California serendipitously is this. So the pathogen is, the slide that I showed before shows you the activity of the pathogen. So the pathogen is active during the spring, okay? That's when everything happens. Now, if bays were not susceptible during the spring, then we wouldn't really have an epidemic, right? Or we would have a much lower epidemic. Unfortunately, those curves here show you both the experimental and the natural. You don't have both, but this is just one trend because, you know, I don't want to overwhelm. And as you can see, the peaks of susceptibility, that's susceptibility, so the larger the peak, the more susceptible the plants are. Bays are most susceptible in the spring. But still, if that's all we had, maybe oaks wouldn't be infected. If, you know, so it happens that the peaks of susceptibility for the oaks is also exactly at the same time. And this is totally serendipitous. Yeah? It really is a coincidence. And it's a coincidence that we call synchrony. So there's a synchrony between the infectivity of the pathogen, susceptibility of the vector, and susceptibility of the dead-end host. And so we think that this really, in theoretical terms, explains a lot of the infection process. What is really important, I already told you this, but how did we get to the determination that the spores didn't move that far? Well, in different ways. For instance, by observing how the infestation was moving, you know, every day. So that's an okay way to do it. However, there's a problem. There's a problem that you don't really know whether what you're seeing, because this is a microbe, so it's not like you have a little, you know, collar with a little, you know, band on the microbe that moves. So you don't really know whether the source of what you know, whether it's a single source that causes the infestation, or maybe there's multiple sources that you didn't know. So that changes a lot the spread rate. So a way that's a little bit, it's different, a little bit safer, it's actually to look at the genetic component. So the genes, and how many of the genes that you find in one point do you find as you move away from that point? Because then you're looking at the genetic makeup. So it's almost like banding, you know. And you have several genes that you look at, and the less number of genes that you see as you go away from the source, it means that the less likely it is that the pathogen can move at that distance. And so it happens that the curve goes down very steeply at about 500 meters. Okay, so basically, it's obviously that's where most of the movement occurs. Between 10, and we know that percentage-wise, most of the spores will be 10, 20 yards, and then it will decrease. But it ends at 500 meters. Already at 500 meters, very few spores will get to the distance, almost nothing. However, there was a big surprise. And the surprise was that, and I'll explain this to you in simple terms. So remember that these spores are really, really heavy because they need to be wet. So guess when a spore is very heavy, and there is a little light wind, you think the light wind is going to pick it up? You need a really strong wind to pick up these spores, right? So you have two patterns. One is the pattern of the spore just falling down. The other one, when you have really strong winds, when a strong wind picks it up, it's not going to release it in a linear fashion. It's going to move it for a certain distance before releasing it because of the speed that it has at the moment that it actually catches it, okay? And so it happens that the second peak that you see there, it's the turbulent movement peak, and that's what explains the movement between one forest and the other. And that range is between 1 and 2 miles. So, And unfortunately, that's perfect because the gaps are almost never bigger than that between the forests. And so now we know a little bit how it moves. And again, because it's turbulent, that is not very regular. It's haphazard or it's unpredictable. While the first part of the curve is more predictable, so we know that that happens more constantly. So, in terms of how do we use this information to help us a little bit, again, if we know that most of the infection happens in the spring, then there's some obvious things, like in areas at risk, try to minimize work in the spring because we don't know how many ways we can help the pathogen to spread. For instance, we've done an experiment showing the wounds make trees 10 times more susceptible, okay, when they're fresh. So, also, if you have, an area that in one part you have the disease and the other you don't, it could be in a single property, never go, especially during the rainy season, from the area that has the disease to the area that doesn't have the disease. But always go the other way around. So always first work in the area without the disease, and then go in the area with the disease. So these are simple precautions that, that will allow you to minimize what we as humans are doing to help the spread. The other thing is, there are some approaches to protect oaks. They only work preventatively. They are expensive because you need to do them every year. So one of the big issues has always been, when do people need to worry about starting these preventative measures? Unfortunately, most people wait until the tree is infected. And then, by definition, that's not preventative anymore. And so then it doesn't work. However, in other cases, people err too much on one side or the other, okay? So if we know the fine distribution at the local scale, then we can make a much better decision depending on how close and how far the disease is. And in fact, oaks are at risk only when they're within 200 yards from a front of infection. So very, very, very limited spatial scale. And again, I don't want to, overwhelm you. The only thing I want to point out here is two. Okay, so first one, that forest patches that are under 50 meters, in diameter of the largest dimension, are not very favorable. And this is important because it's kind of one of the traits that tells us the organism doesn't like the light very much. It likes the shade. It's probably very susceptible to UV light, which we could test. We haven't done it yet. The other one is that high diversity actually slows down high diversity in terms of composition. It's something you will like, I'm sure. Slows down the disease. But this is interesting because one of the first papers that came out said the opposite. Said actually highly diverse forests were spreading the disease more. Why? Because it was the definition of diversity. In the first paper, the definition of and it was not one of my papers. The definition of diversity was bay laurel oak. So the more diverse it was, and the second one is a true definition of diversity, which is the number of species. And it actually turns out that if you don't just focus on bay laurel and oak, if you focus on all the dominant, codominant tree species, the more you have, the slower seems to be the spread of the disease. So that's a little bit good news, but unfortunately, okay. So one of the things that we've been recommended that are very, very powerful is try to eliminate the vector in the way that makes sense. So we've had people go out there and try to get rid of all the bay laurels, and of course a year later they have 10 times more bay laurel than they had to start with. So that's not really the approach. So the approach that, well, in a managed forest, you may think of maybe prescribed burns. I mean, especially if you're away from urban areas. In urban areas, what we'd be recommending is remove the bay laurels from where they could infect oaks that you want to protect. Okay, so basically this is an oak by oak approach. You pick, people come to me and tell me, I have 4 oaks I want to protect. I tell them to always cut it in half, I just say it's much better for you to focus on less and do it well than trying to do as much as you want and then do it badly, unfortunately. So it's very imperfect, but we finished a very long study and we've shown that it actually kind of works. This study was done by clearing bay laurels for 10 or 20 meters and then looking at how many spores we were finding. And every time you see a red, and this is just, every time you see a red, there's a potential infection for oak, okay? So you can see that if you remove the bays, the potential infection for oak goes down really dramatically, okay? However, it doesn't, the spores don't disappear. So if we had very wet years, that probability would go up. And so the removal of bay laurels is Usually, I'll skip this. It's associated with something else that increases the defense mechanism of the tree. Why? Because if a lot of rain comes, more spores arrive, but the tree is slightly more resistant. The two together should actually protect the oak from becoming infected. So we always recommend them together. So we've been talking about oak-centric approach. So this is a very limited approach. There is a stand-wide approach for people that are managing the forest, not really in the Bay Area. So it's a little bit more complicated. There is also a narrow one, a broad one, that it's the so-called minimum effort, maximum output approach, which is using the cycles of the disease to our advantage. So we completed a study in San Mateo County, which it doesn't mean that it's exactly the same thing is going to be true in the rest of California, where we found out that after 2 years of drought, we found out two very interesting things. The disease is a little bit everywhere in all these plant species, on the soil and the water, but after a dry season and after a dry year, it always comes out of bay laurel leaves. So bay laurel leaves are actually the reservoirs. So the other ones are not significant, which we didn't know. We thought the soil may play a role. And we found out that after 2 years of drought, only 5% of the bay laurels carried infection. So this approach would be, if we can figure out where the 5% is, that's actually a number that we can deal with at the forest level. We can actually take out 5% of the tree. That's an approach where we're not trying to get rid of the disease because we can't, but we go from an epidemic level to a chronic level. And obviously with mortality being much less. And this is kind of the patterns that we see even within a year. Okay, so as you can see, the number of trees that carry the disease during the dry season within a year goes down in the fall, but during the drought that gets compounded by the fact that it's dry, and so that's kind of the principle behind it. Bay laurel removal, strengthening the response of the tree. So the compounds that we tested are called phosphonates. It's the simplest compound you can think of is phosphorus, linked to potassium or sodium depending on the manufacturer. You could drink it. In fact, in Australia, where this thing was basically invented, they drink it to show the people that want to sell it, they drink it to show that it's totally innocuous. Of course, then they have all sorts of problems, but no, actually, the product really is innocuous. And what does it do? So really, what it does, it does a lot of things. It's a black box. Okay, we don't really fully understand. Two of the most important things that it does. One, it thickens the cell walls, so it makes it harder for the pathogen to kill the cambium, because they're more lignified. There's more lignin in the cell walls. Number two, and maybe more important, I don't know many of you, even if you're CNPS, do you know how plants respond, how plants defend themselves from disease? Do you have an idea how it works? Do they have an immune system? Do plants have an immune system? No, even if it's called, when I was a student, we couldn't call it an immune response because people would go crazy. Now it's called an immune response. But it's really a system of signaling, receptors and elicitors. Okay, so the elicitor is a signal that's produced by the pathogen. The receptor is the ability of the plant to receive the signal. Based upon the receipt of the signal, the plant produces compounds that will kill the pathogen, okay? Or they will kill the cell of the plant itself to stop the pathogen in its growth if it's a biotroph. So this is kind of, in simple terms, how plants defend themselves. Where is the key step in the defense mechanism is the recognition process. Why does it not work for sudden of death? Because our plants have never seen sudden of death before, so there's no way they can recognize it. So what this compound does gives the first block that's necessary to produce these antibiotics, these secondary metabolites. And when the plant has a certain amount of these blocks, it says, what the hell, I'll produce the antibiotics. It doesn't do it normally, otherwise ecologically it's not competitive. So this is the principle. The problem is, and it's a funny story, when at the beginning of it's a very funny story, I was just been hired junior faculty at Berkeley, I was scared like hell and found this organism, was testifying in front of Congress every other day. Then, I go into the treatment and, we see that this works, and then people from the world of human medicine, big names, come to me. We have these big meetings, and the first thing that they say, Matteo, we're going to sequence the entire, remember, it was the time when the human genome had just come out, And so the Department of Energy had all these machines we didn't know what to do. And sudden death was one of the first, if not the first, organism in the first to be sequenced after the human genome in these machines. And it was absolutely ridiculous because we didn't know nothing about this organism. I mean, at that point, the genome sequencing was something very complex. And nobody was only done for model systems, so systems that you knew everything about. Basically, we just had isolated the organism like 2 months before, and it was selected as, for sequencing of the entire genome. So from first isolation ever and completion of the sequencing was 3 years, which I think it's still for a eukaryote is still so these people were saying, well, once you have the genome, you can cure all the trees. It's interesting how people, from that world, they have no understanding whatsoever. And I'm like, wait a minute, guy. I remember, because I was very nervous because these were really big people, right? Big names. I was like, but there's a little problem because, like, for instance, trees don't come to see the doctor. So just first problem. Second problem, we can't play nurse and doctor with trees and inject them very easily because we don't really. It doesn't work that way. So there were, there's many. And of course, you understand that many other issues with, how do we treat trees. And the one that was of interest to me is how do you deliver the product? So that was, so to be honest, it's still very imperfect. So you can either inject it in the tree, and now after 5 years of recommended injections, we found out that there's a little bit of damage, so we have to tweak, the injection before we recommend it again. It's inevitable. And the other way is to apply it with a surfactant, so it goes through the bark. Also very inefficient because anything comes in between you and the surface where you apply it, it's going to decrease the efficacy. So anyway, so that is the problems that we're facing. However, there are ways that are, borderline, but they're effective. They deliver the product, and they can be used for treatment. And again, we're trying on Tanoak, even if we know that really for Tanoak it won't work. And is that just, this gives you an idea after a few years, I don't know, of in the treated, after 3 years in the treated, 75% of the plants were healthy versus 56% were healthy, okay? So this is 36 plots, it's pretty large numbers, about 1,000 trees. So it's, it seems to be having an effect on Tanoaks, which is, good because Tanoaks are so susceptible. I don't see this as preventing mortality of Tanoak, but I see maybe as extending their lives. So the issue is going to be, will it work for 10? When will that number, change? But if it does that for Tanoaks, then it means that for oaks, it would be a lot. Obviously the efficacy would be much more. Again, it's Berkeley and it's me and I'm crazy. So when UC Davis does the same type of study, they go with ATVs. So our colleagues at Davis that were doing the study, so they have these ATVs that go in the forest. Instead, I found a photo of the Ho Chi Minh Trail with the Viet Congs, and I modified the bicycles to carry really heavy amounts of phosphonate to treat the trees in the forests. So you have to do that in 32 sites. Again, I lost all of my other students. So I love because so basically, a few things about the treatments in general. First of all, really right now, and it's, I know some of you probably are doctors or scientists, and so I know that it's a little bit unsatisfying that we are recommending two things together, but we can't really assess which one is working better than the other one. It's not perfect, but we know that both have a little bit of a component. And because there's so much effect of the weather conditions and the ecology, it may be that in one case one is much more important than the other, but together they seem to cover a little bit more. The other thing that is important to keep in mind is that the effect of the treatment is on the plant, not on the pathogen. So it's the plant that does its business. And so there is time required for the plant to respond. And if you treat in the spring, there may not be enough time for the plant to respond because in the spring that's when infections are. We don't recommend treatment in the winter. We don't recommend it at all in the summertime because the trees are water starved, they're not active, so they don't respond. Okay, and also in the winter for a small period of time. So the best time for treatments, again, in this very general overview, is the fall. Because in the fall, what happens, the temperatures at a certain point will drop. And so even if it doesn't rain, the water that was insufficient before because of the high temperatures, all of a sudden becomes more than sufficient. As a result of that, the functions that we tested this, so we looked at translocation of things, of carbohydrates and so forth, in the plant goes up exponentially. That is when is really the best time to treat, because the plant is active and it gives enough time for the plant to build up whatever it can do. The other good news with the organism, and again, I don't want to go crazy is I will, for many of you have gone through the pine pitch canker epidemic, right? So you remember, and still now, it's ongoing. And the big deal with the pine pitch canker is that it produces these microscopic spores that are sticky. And so the tools, if they're not cleaned really well, they will spread the disease, very significantly. It's completely opposite for Phytophthora ramorum. That's why we don't really think that people have moved it necessarily many times. So the spores of Phytophthora ramorum are always embedded in organic matter, in debris, in plant tissue. So the consequences of that is that if you clean the tools so that they look clean, actually, really, you have basically eliminated 99.9% of the spores. I sound like one of those commercials. This lies on 99.9%. And so really the most important step is to ensure that there is no soil and no plant debris on the not just the tools, but also the tires of vehicles and so forth. So that's kind of so that's relatively good news. Okay. Okay, the last few slides, I hope that I'm not going overboard here, but this is obviously the most important thing. I painted this scenario of lack of resistance. Unfortunately, it seems true, but you cannot say until you test it. So it's not an easy job to do, because when you test plants that grow in different forests, as you know, the range of the disease is very broad, you're actually testing two things at the same time. One is actually the ability of the plant to genetically respond to the disease. And the other one is the effect of the location where the plant is growing, that's having both on the plant and on the disease. So basically you cannot disentangle the two. It's an okay first step, but it's not enough to understand whether there really is resistance or not to the disease. So the way to do it is actually to collect acorns from all the different locations, and then you have to grow all of the plants in the same location from seed or from acorns, and that's called a common garden experiment. And this is also a good story because, so our common garden experiment has 22,000 Tanoaks. And so when I was all proud, so we actually built with our hands a whole house at Berkeley to put this huge number of plants. So when I was like, to them, so, people from, the other side of things, 22,000. We test 2 million Arabidopsis every year. So I said, okay, come here. Arabidopsis after 6 months, like this. Tanoak after 6 months, like this. So, it's not the same thing. After 2 years, Tanoaks are 10 feet tall. Then, real big, it's a complication when you have 22,000 plants. So, at this point, we tested 22,000 seedlings or acorns from about, I think, because the data I'll show it's old, about 400 mother trees. These are acorn produced on the trees themselves, so they're mostly half sibs. They're only 50% related to the mother because they're openly pollinated. Okay, and this is so this is still 2030. So this is from a few years ago. This is how the process works. So we go to the mother trees, we climb the trees, we collect the acorns. We don't collect from the ground. We stratify the acorns, so put them for a cold period. We grow them in the greenhouse. And at first we didn't know, but after 1 year we tested, we challenged them with the pathogen, and we challenged a large number of acorns from the same mother, and then we determined how many of the acorns from one mother did okay, or did not okay. So we quantified their response to the disease. The ones that are promising go to a secret location in Monterey County where there's tons of disease that are planted in the wild, and then we see whether they actually do okay in the wild. Okay? So what we found out through this process was that when we tested seedlings that were 1 year old, the result was meaningless. So The 1-year-old tests were not really very informative, but the 2-year tests were. Okay, so this is, and again, that was just pure luck, because at one point we didn't have time to do all the tests. We said, okay, we'll do the other ones next year. So all the families that we tested after 2 years, the results were significant, and the ones that we tested the first year, they were insignificant, because the plant has not built up, and that's a common thing. So what we started seeing for some families was, basically, we had a small brown bar, and we had a small red bar. That means that mortality in the field was matching susceptibility in the lab. We did not find any resistance. Okay, so no resistance, we expected that. So what we're talking about here is tolerance. So it means that it's more difficult to infect the plant, and once infected the plant, the lesion in the plant develops more slowly. Now, these two traits can actually save a tree for 2 reasons. If the tree grows faster than the rate of growth of the lesion, the tree will survive. Okay, so because basically the pathogen cannot keep up. The other one is that if the tree is naturally challenged by the pathogen in a year where conditions are not ideal, or in an area that is a little bit on the fringes of what the ideal conditions are, if the tree has the genes that allow it to be a little bit more tolerant, then potentially it won't become infected. So it does have a practical implication. So it can actually be used in theory. It could be used. And so we asked the question: is it just theory or is it true? And we're just coming out with a paper on a nice journal called Evolutionary Applications, where we've shown that if we use our lab assay to pick the families, we get the maximum survival after 5 years in the field. Okay, so we know that it's not a perfect correlation, right? But overall, we get 100%. And we learned a lot of things that were very interesting. That we learned that it's better not to put all your eggs in one basket. So we learned that if you pick all of the trees for their resistance, for their tolerance to the disease, that's not so good. You also have to pick trees that are ecologically fit. So both things are still important. Even if you have a lot of pressure of the disease, the tree needs to be able to compete with the other ones for other traits. So a combination of the two is the one that maximizes the results. So this is promising. Of course, people told, oh, but this is just seedlings. And I told them, yeah, so give me 100 years and then I'll tell you the results. Or give me a time machine and then I'll tell you what happens with the results. So unfortunately, it's all, we don't have a time machine. It's Berkeley, but we don't have a time machine. Maybe Stanford has one. So that's kind of what's going on right now. And even if we go back, we thought of ways to find out whether the adult, we can go back to the mothers, but it's different because these are acorns that are these are recombinant. So going back to the mothers will not tell us the same thing. So we can't fully answer the question, once they're adult, will this tolerance still be there or not? However, if we plant seedlings that are all going to die, they never get to be adults. At least in this case, we know they will get to be adults, and then we at least we have more time to deal with the issue. So we talked about that. So we did a lot of research in my lab. We really focused a lot on the plants. That's why I'm happy to be with you. And it's considered a bit unusual for a plant pathologist. So we did a lot of work on the plants, really just the plant side of things, because we were interested in finding out how do the different populations of these native trees in California respond. And we had a botanist working with us to do the genetics on these plants, so that we tried to associate the genetic structure of these populations with the phenotype of the response to the infection. And we found out some really interesting things. And I'll give you a quick example. Do you remember that early on, with sudden death, Chinacamp State Park was the big talk of the town. Every time there was a front page of the paper was because it was kind of the poster child of sudden death. And I don't know how many of you know Chinacamp, but Chinacamp we never thought it was really an ideal place for sudden death because it gets really hot. And it's really borderline. Okay, and for instance, when we compared Taylor State Park, whatever, the Redwood, what is it called, Taylor? Samuel P. Taylor, thank you, sorry. Samuel P. Taylor. And Chinacamp, the infestation of Samuel P. Taylor was a lot lower than in Chinacamp, and we never could figure it out. Then we did a study on how good were the bailors in carrying the infection. And it turns out that China Camp beats every single other location in the state of California and Oregon. So there is a huge role that's played by the we call it vector, but because it can actually make up for the lack of perfect environment. So now we have, actually we actually let us publish it. We kind of put our results from a lot of populations together with a little bit of climate data, and so we estimated what the risk is for different locations, and that kind of, we are using it as kind of a relative roadmap to predict what's going to happen, right? So if we have very susceptible bay laurel populations, we know it's going to be bad, no matter what, and vice versa. So it's something that all the other models were simply based on the weather. They could not get to that because we didn't know how the hosts were collaborating. So, in the end, when everything is said and done, because you all will come to the sublets, I know, you have your boots on already, you'll come to the sublets, and you will participate in this wonderful, they really are wonderful. Really one of the most useful things that we can do at this point is just let people know, and also keep it in the public eye, and let people know the disease is moving, you know, and also not let people fall prey of people that want to sell them something all the time. And so the maps can be used to say, no, I'm not going to buy this for you because the disease is 5 miles away, and we very well know that it's not really a risk. You know, I keep my eyes open. And the maps that we generate are Google Earth maps with these icons. The yellow ones are not there anymore, but the green and the red means infected, the green means not infected. And we have learned a lot of stuff from the citizen surveys. We have learned that in California we have 2 completely different areas in terms of disease development. So the truly coastal area, in the truly coastal area, once the disease becomes established, that's kind of it. But inland, meaning like here, for instance, we consider inland, central Sonoma, we see huge variations following the climatic pattern. So as it gets drier, the level of infection drops about 3 times. So all of a sudden we have, we know now that in these interior areas, we actually have more chances of working with the climate to actually slow down the disease, but probably that's not going to be possible on the coast. We found a lot of new infestations, including one in Atherton, which is my pride and joy. One in Atherton that causes, this huge fight among the neighbors. But then eventually the good neighbors won, they cut down the bay laurel, and for 2 years now the infestation is not there. Atherton was the perfect case because it was several miles from the continuous infestation on the slopes of the Santa Cruz Mountains. So that was really a good situation to try something like that. And we can't say success because we have to wait a few more years, but I think it was a good thing. And the third thing that we learned last year, I don't know if you remember reading, but the detention span of us in this century is very short. My relatives in Mexico and in Italy, they tell stories, still remember all the details. There's a big oral tradition that we don't have in this country. So, what was shared with the public last year was that a very interesting phenomenon, which affects you and affects the East Bay. So what we reported was that, and here it's not so surprising, that the disease spread with very high levels of infection and infestation as it was moving on Skyline. And then as it moved east, and it came from the south, so as it went north and east, it slowed down because the conditions become less favorable, it becomes dry. That was not a big deal. But the big deal was in the East Bay, because in the East Bay it was a mirror process. So the disease was introduced, we think, as early as it was introduced in Marin County. But because it was introduced in Contra Costa County, where it gets pretty hot in the summertime, it moved really slowly. And so last year, 2 years ago, when the disease arrived on the edge of the Oakland-Berkeley Hills, I made the statement, don't worry, because it took 20 years to get here. It will take 20 years to go into the neighborhoods. Boy, was I wrong. So what happened is that the weather changes completely. The climate changes completely. The climate changes completely on the top, because then you get into the fog zone. And in 1 year, the disease actually went 10 or 15 times more. The infestation rate accelerated 10, 15 times. So this, all these things we could have maybe predicted, but we really didn't understand how sensitive it is to these slight changes, so that on the east side of the Berkeley Hills, it moved very slowly, and now on the west side, it really moved fast. So the opposite of what's happening here. So we learned this through the volunteers. And then we have these, the concept behind the maps is that if you are within 200 meters from a positive tree, there's high risk. If you're a kilometer from a positive tree, the risk is moderate. But if you're more than a kilometer, the risk is low. Okay, so this is for oak infection. So this is very, very, very useful. And if you come to the training, we'll show you the fantastic app that we generated, which is the most amazing app. It shows you all the database, the distribution of the disease, and you push a button and we calculate the risk of the location where you're standing, because it calculates all the trees that have been ever sampled, whether they're positive and negative. It's really, really cool. And finally, it's limited, of course, because it's limited by the database, but it's a non-subjective way of deciding or giving help to local people to decide, should I listen to this guy or should I, follow the science? And they can do it even if they don't know all the stuff that you know now. The SawMap Mobile is based on the SawMap project. And the SawMap is the same thing as the map that I showed you before. You go to my website, and if I invite you to go and to actually look at it, to see, to kind of learn the distribution of the disease in your area. You will download it, and it's very intuitive. And you will be able to if you click on the icons, it will tell you some information. The only caveat I have to give you is that it's a very data-heavy map, and so a lot of the data tend to stack on top of each other. So you need to enlarge as much as you can, and then you also need to click. And if you click on some icons, they will actually open in a rosette of all the icons that are stacked on top of each other. Okay, so that's the only caveat. With that, it's very self-explanatory. And the cool story about the SawMap is that the SawMap started, the SawMap is kind of an afterthought of the sawblitz data, okay? And the sawblitz data is funny because when we started the sawblitzes, I think there's a sawblitz organizer here, she knows that. We all, the organizer, we said, the data is going to be worthless. But the education is great, right? And then people go in the field and they see the disease. And so we said, so let's not worry about the data. It's expensive. I had to raise about $200,000 to run it. And so the funding agencies were like, okay, the education is great, go ahead and do it. So then after the second year, we look at the data and say, oh my God, I remember saying, this is kick-ass. So the data are actually amazing. They're really, really good. We run analysis. We saw that people that go through the training provide the same quality of data throughout the state. So we get really good data. Once we started having 5,000, 6,000 points, we said, wow, this is a lot, And so what actually happened, which has never happened before, the government decided to pool their data with the volunteers. So they gave us their data, and even more difficult than the government, all the individual researchers, I had something to do with that. So all the individual researchers gave us their data even before they're published. So basically we have all the data that's almost ever been generated for sudden or death, and we have it in the SawMap. And being of Italian ancestry, the cement shoes at the bottom of the bay work really well. So those that didn't want to give us the data ended up like that. Okay, so so this is a good story of a volunteer. Not only empowers the community because then the community is informed, and we noticed that they were doing a lot more in terms of disease management. Not only it's wonderful because 500 people provide data for 1 million people, but even more because if you think about the residents. But it's also fantastic because it generated this incredible distribution of the disease with no equal in the world for any other disease. So it's the only disease that has this really fine scale distribution. You're all tired, so I'm kind of going to skip the fire thing, but there was a big fire if you remember, in Big Sur, and so that was used by the Davis group to figure out the effect of fire. Make a long story short, does fire kill Phytophthora ramorum? Yes, if it hits it, but unfortunately fires are not all over the site, so in every single site, the disease came back a year after. Not the year of the fire, but a year later the disease came back. Okay, so fire, and we knew that, despite a paper that was published early on that was really wrong. Fire doesn't really have it has a direct but very transient effect on the disease. Fire could have an effect on the epidemic if we reduce the density of the forest and if we reduce the component of bay. So yes, that way it would have an effect. But On the disease itself, it didn't have an effect. The other point of fire that I wanted to share with you, that where you had the fire and there was sudden death, guess what happened? Medium-sized redwoods were dead. Why? Because the fires were so hot, they were too hot, not for large-sized redwoods, but for medium-sized, and we're pretty big, 40 to 70 centimeters diameter. So that's a huge problem, because think how much is invested on those trees. So you're losing what you invested in the past and what these trees were going to do for the future. So that's one of the consequences. The other consequence of fire is that for about 2 years, you have the potential for these hot spots, because the trees are standing with all the dry trees standing. But then the tree falls down very rapidly, and the danger ends. So I had some final remarks, because I think that this is not just about a disease, about the biology, the ecology, although we are very concerned about it. And these are the points that I wanted to make. The first one is how, sudden death really is the child of really poor regulation, poor policy, of a badly organized trade for the environment. Because it's a trade that is based on movement, intercontinental movement of plants to maintain an old system that's very advantageous to them. But it is with huge consequences for the environment. Sudden death is not the first one. Almost every big, at least 80% of the big exotic disease has been introduced through plants that were traded, okay? So chestnut blight was introduced that way, cypress canker was introduced that way, so on. So, at what point can we exert enough pressure to say, okay, let's change the system, okay? Let's stop allowing for movement of plants in between continents, but even in between regions of a continent. In particular, because there is no connection between an ornamental rhododendron and the forest, meaning that these were not plants used for reforestation projects, okay? These were plants used for aesthetics, which is wonderful, I don't have a problem with it. But, so, there was no risk-taking in favor of the forest. The risk-taking was all in favor of the companies that make the bucks. So it's a social responsibility, and awareness that we need to increase with politicians and with the public. Big problem for us, the biggest problem we have, that's very big, but we may never solve it. Maybe we'll solve the second one. So you know what happens when you have a very dangerous organism? You have a thing that's called quarantine, you have a thing that's called zone of infestation. What does it mean? It means that the government decides that they cannot do anything about the zone of infestation, so all of the policy is designed to protect the rest of the country from moving the organism, from the zone of infestation to the other parts of the country. There is a problem, because in California, the nurseries have these very different groups of strains. These are different in dramatic ways, different sexes. So if the other sex were to be introduced, then the organism could actually reproduce sexually, which means accelerate the evolution incredibly. They can survive on much hotter temperatures and colder temperatures. So you tell me what that does. But the government is now protecting the 14 counties in California because the law doesn't allow it. Even if genetically the strains are so different because they have the same specific name, the government says is powerless, even if they know very well that these are completely different organisms. Third point, why is such a problem if it doesn't move very much, right, by itself? And so I explained how it is very infectious and it reproduces very, very rapidly and a lot. The other thing that I wanted to bring at the end is this. When I started working on this organism, I had, for about 2 or 3 months, I was really in a bad mood because I was like, I think this is going to end the world. No, really. I mean, it could infect everything. I mean, I said, this is it. I said, and I really am sorry that my name is associated with it. I said, I really am not happy about it because it's going to end the world. Well, naive, but you're entangled in these thoughts. The good thing is that really the organism is extremely susceptible and limited by the different ecological conditions. Dryness, sunlight, gaps. So this is really kind of the saving grace. And potentially this is where we need to move in terms of keeping the infection level down. Okay, so That's why, you know, it hasn't killed everything, because it's very strongly modulated by these ecological factors, also including the other Phytophthora species that occupy the same niche. So the ones that occupy bay laurel leaves, we know that, for instance, in dry conditions, they match ramorum. So there is a role that's played by those. And that's the good news. The last point is, as of 2012, so people tell me, oh, silent death is not a problem anymore. As of 2012, as I told you earlier on, only 15% of the habitat that can be colonized has been colonized. So we are at the beginning, not at the end. You know, we hope that it's not going to go to 100%. We also hope that the organism is not going to change, and I'll give you a quick example that you should be aware of. Sudden death problem with oaks, it kills oaks, and it spreads via rhododendrons. Okay, well, there were two other releases, one in Belgium and one in the UK, and it killed 10 plants in each state, because it wasn't really working like in California with bay laurels and oaks. It was less effective. 3 years ago, the organism encountered larch. Now, you are botanists. The most distant species that I could think of from these that are evergreen broadleaf trees would be deciduous conifer. Yeah. On larch is incredibly infectious, incredibly susceptible. Bottom line, they cannot plant larch anymore in the UK and in Ireland. On the west side, at least where it's wet. We cannot. So that's the same organism, and they call it southern larch. So the concern is, you know, the hope is, and you know, again, I don't want to do doom and gloom, and maybe it will never happen. However, we can't be complacent about, oh, it's here, you know, we don't care. We should be very, very cautious. And this brings me to the last slide, which is what happened last year when the disease arrived in massive force in Golden Gate Park. An entire sector of Golden Gate Park is infected. It's only 3 trees, but it's 3 big bay laurel trees that are infected in Golden Gate Park. And when I went to the managers, I said, guys, the main concern here, well, one concern is that people may bring it back to Australia because they don't think about the disease, or New Zealand, or, you know, or Europe because they have a different groups of strain. Number 2 is that basically we have the most exotic population of trees in North America here, the most weird combinations. This is a wonderful testing ground for the organism to develop something new. And so we don't want to really try our luck too much. And so, you know, these are the kind of things that may be a little bit difficult to understand. So, I wanted to, first of all, thank you for your patience. I know I went a little bit over time. I wanted to thank the funders because I have been incredibly lucky with my funding support. I have an incredibly well-funded lab, equally by 3 agencies, actually the USDA Forest Service, the National Science Foundation, NIH, and the Gordon and Betty Moore Foundation, which are local people, so I'm sure that you're aware of them. So they give us millions and millions of dollars to do the research. And I invite you to go to the website. It's a little hooky, but it's official. It's matteolab.org. You will find a lot of references, and in particular, you will find a review that we wrote for a journal, but I wrote it in kind of colloquial terms for eukaryotic cell that will bring all of these things together in a nice way. It's by Garbelotto and Hayden, and it's in my website. You go to recent publications, you can download it. So the other website that you should be aware of is www.suddendeath.org. So these are 2 websites with a lot of information. And I hope to see you at the Sawblitz in Montalvo. If I don't see you, I know where you are. And expect to meet you. And thank you very much. And